So, as I mentioned before, my pointer here, the way we classify viruses is the first step is, do they have DNA or RNA as their genome, and is it single-stranded or double-stranded? And we seem to find the greatest variety of these of RNA viruses in animals. Many viruses that infect animals also have an envelope, and that is the third component that not all viruses have. This membranous envelope is stolen from the cells they infect. Sometimes it's stolen from the outer plasma membrane of the cells. Sometimes it's stolen from the nuclear envelope and the Golgi apparatus membrane, but it is made from phospholipids that are synthesized in the host cell. Viral genomes do not contain genes for synthesizing these lipids or phospholipids. They obtain them from the cells in which they infect. So here's what happens with an enveloped protein. So we have the genome, the capsid, and then the fatty envelope. And the proteins that are sticking out of that fatty envelope are in fact viral proteins, because remember, it has to have those proteins on the outside to stick to the host cell. So one of these enveloped viruses sticks to the receptor of whatever kind it's binding to the host cell, then it enters the cell. It generally loses its membrane on its way in, then it disassembles just like other viruses do. Now we have our viral genome, which sometimes this RNA will behave like a messenger RNA for the capsid proteins already. Sometimes it has to be copied to another RNA. This is how the coronavirus does it. The RNA genome is copied into another RNA, and it actually brings its own enzyme with it to do that, because there isn't a host cell that has that. The messenger RNA then goes to the ribosomes to make the capsid proteins. There is also proteins that are going to end up on the surface of the cell that are viral proteins. So two kinds of proteins are being made: capsid proteins, but also these surface proteins. And they're going to go through the Golgi and end up on the surface of the cell. So when the virus then leaves the cell, it's going to take that membrane with it that has its own viral proteins studded in the outer membrane of the cell. So retroviruses are interesting, and they've been known about for many decades, and they were discovered to cause cancer in chickens first. And now there are several bird cancers that are known to be caused by retroviruses. And there's also a couple of human cancers that are suspected to be caused by retroviruses, rare ones though. HIV was the first retrovirus that was discovered to cause a significant disease in humans, and that is AIDS. So HIV is the name of the virus, human immunodeficiency virus, because what it does is it destroys one particular part of the immune system. So immunodeficiency. The disease it causes is AIDS, acquired immunodeficiency syndrome. So HIV is a retrovirus. It's an enveloped virus. So here's HIV, and the receptor that it binds to is only found on one particular subset of human cells, white blood cells that are the CD4 cells. That's the receptor that's on the surface. So that's what they stick to. They enter the cell. They have to bring one of their own enzymes with them, reverse transcriptase. Because they're an RNA genome that's going to be converted into DNA. Now, no living cell has an enzyme that can convert RNA into DNA. What cells do is convert DNA into RNA. That's transcription, right? That's what happens during gene expression. To go from RNA to DNA takes a different enzyme, reverse transcriptase, and the viruses have to bring it along with them because no living cell has such an enzyme. So they bring along their reverse transcriptase that transcribes the viral genome into DNA. It inserts itself into the nuclear genome randomly, which could disrupt genes. then the genes that are in there are expressed just like any DNA gene. Messenger RNAs are made that code for both the capsid proteins and the surface proteins that are going to end up sticking out. The surface proteins have to go through the Golgi, and then they end up on the surface of the cell. Once the virus particles are assembled, then they are released. They steal membrane on their way out, and that membrane is already studded with their own viral proteins. So this process will eventually destroy this cell. If this virus is replicating inside of this cell, it's stealing membrane, it's hijacking the manufacturing of that cell. This cell is eventually going to lyse and die. So here's some nice scanning electron these are TEM images, sorry, transmission electron microscope images of an HIV particle entering a cell. And of course, these are not like video still shots. This is different slides that just happen to capture different parts of the process. So they look like they're sequential, but it's not. These are from different events because obviously everything is sliced and dead to look at it under the electron microscope. Okay, so here's a particle that's stuck to the surface of a cell but hasn't entered yet. Here is it being taken into the cell, and here we have a virus particle leaving the cell. So it's stealing host membrane on its way out. So once it inserts itself into the genome of the host, then we call it a provirus because it's not really acting as a virus anymore. It's in the host genome. And just like, well, for the HIV, once it's inserted, it can stay there. If it doesn't rupture the cell and the cell continues to divide, it's going to make copies of that virus. So the RNA that's made can behave both as a messenger RNA for synthesis and also be copies of the new RNA genome that's going to be released from the cell. So where did HIV come from? It's a new disease. It was first noticed by science in 1981, and it was only noticed because a few doctors had multiple patients with the same assortment of weird disorders. Young men who were healthy suddenly had a rare type of fungal pneumonia, which is only usually seen in people who are severely immunocompromised, a type of rare skin cancer that's usually only seen in the elderly, Kaposi Sarcoma, which now we know is caused by a herpes virus, but that was not known in 1981, and chronic weight loss. And then they would eventually succumb within a couple of years of being diagnosed. And some of the patients knew each other, and some of the patients were sex partners. So these doctors reported it to the CDC that they thought they possibly had discovered a new disease that was contagious, that caused catastrophic collapse of the immune system, and might be sexually transmitted. It didn't take very long for the virus to be isolated and discovered, a few years. Since then, since it was first identified and was traced back to the big outbreak that was identified, at least 35 million people have died from AIDS. That is the low estimate. That's the number of confirmed cases. The actual number could be 20 times that high. Some people put the estimate as high as 600 million people may have died from AIDS because so many people have died they die of other things. They die of tuberculosis. They die of pneumonia that doesn't get recorded as a death from AIDS if they never had a test for the virus. And it's rather difficult to get. So HIV is not something that you're going to get by somebody coughing on you or somebody touching you. It has to be transmitted by blood. So some of the infected person's blood has to get into your blood. So that's why dirty needles has been a common mode of transmission, as well as in the 80s, blood transfusions. transmitted the virus. Also, dirty and improperly sterilized hospital surgical equipment and dental equipment has caused the transmission of AIDS. And it also can be transmitted by sexual contact, heterosexual and homosexual contact. But even that is difficult because there still has to be blood-to-blood transmission in order to transmit the disease. So where did it come from? That's been a question. Where did this new disease come from? And the obvious answer is that it probably jumped the species barrier and came from an animal. It's a zoonotic condition or a zoonotic disease. So earlier cases were sought, and they figured, well, you know, if a doctor had one patient who had Kaposi sarcoma or fungal pneumonia, and it was just one person, it would just be a mystery case that they couldn't solve. And so the CDC surveyed doctors around the country and asked them, have you ever had a patient in your career who died mysteriously, who maybe was a young person who had Kaposi sarcoma, fungal pneumonia, weight loss, a catastrophic loss of their immune system for no apparent reason? Have you ever had anybody like that? And in fact, they found cases going back to the 1960s in the United States, just like one here, one there, one here, one there. So this disease, AIDS, had been percolating around in the US population at extremely low numbers since the 1960s. So if it's a zoonotic disease, something that recently jumped the species barrier, where did it come from? So once it's percolating in the human population and being transmitted continuously in the human population, now it becomes an endemic human virus, which it is now because it's now being continuously transmitted in the human population. But it wasn't at some point. It's a recent virus. So where did it come from? One of the ways we can trace viruses, and this is in fact this method is being used already with tracing the new coronavirus that we're dealing with, is the molecular clock method. So viruses mutate at a very rapid rate, and the molecular clock method takes advantage of the fact that these mutations occur at a fairly regular rate. There's a ticking of the clock, an accumulation of mutations. So if you take a sample of virus from two different people and you compare them, the more mutations there are that are different between those two viruses, the longer ago those viruses were in the same person. It's like you could create a little family tree of all the viruses that cause one particular disease. And so if you do that with HIV, if you take the viruses that are existing today, all of the HIV viruses that exist today, we actually can learn a lot about when this virus may have originated and where it originated. So errors are made in every person, every single person. Once that person transmits it to another person, there's going to have been mutations already. So every time the virus replicates, there's a chance for errors. So when they compared all of the patients in the US and sequenced their viruses, figured out the sequence of the RNA genomes, and created a little family tree and asked, when did all of these viruses unite in a single virus? The year that they got was 1966. So that implies that the first US case was around 1966, and then all the other cases that are in the US were directly a result of that first case back in 1966. And we don't know who that person was. We don't have a United States case zero for that. The so-called patient zero that they talked about 20 years ago, we now know that individual was not, in fact, patient zero, but probably 20 years, or not quite 20 years, 15 years after patient zero. When we compare sequences from around the world, what scientists discovered was that the virus that most closely matches the US versions of HIV are versions of HIV from Haiti, which is close by in the Caribbean. So most likely mode of transmission to the U.S. was from Haiti, and not from Haitian immigrants coming to the U.S., but United States citizens going to Haiti on vacation. There was a lot of tourism in the 1960s from the United States to Haiti. So that's probably how the virus entered the US from Haiti. When we compare viruses in Haiti to viruses around the world, the closest matches are in Africa. In fact, there's several reasons to think that Africa is the origin of this virus. So how do we know that? Okay, so when they compare HIV viruses from around the world, HIV in Africa has the greatest genetic diversity. And for anything, wherever that organism has the greatest genetic diversity is probably close to where it originated. As things migrate around the planet, they lose genetic diversity. So there are 4 varieties of HIV, HIV 1, 2, 3, and 4. Only 1 and 2 are found outside of Africa, and most of the rest of the world only has HIV 1. HIV 2 is second most common, and then HIV 3 and 4 are only found in Africa, on the continent of Africa. So that seems to indicate that Africa is the origin of this virus. So they did the same thing in Africa that they did in the United States. They asked doctors at the big hospitals in many different countries, do you know of any patients who may have died mysteriously from a collapse of their immune system, mysterious weight loss, anything like that from earlier than 1979? And in fact, they found tissue samples from 2 patients that a doctor had saved, one who died in 1959 and one who died in 1960 in the same hospital in Kinshasa, which is in West Africa, that both tested positive for HIV-1. So these are the oldest confirmed cases of HIV-1. So we can ask the same question of these 2 samples. You can do sequence the virus in both of these and ask the same question. Okay, if we put those in our little viral family tree, how long ago were those 2 viruses in one person? And the answer is 1908. So HIV-1 originated around 1908, and there was only a single infected person in 1908. And that person passed it on to others. So where did it come from, though? If it jumped the species barrier, what species did it come from? And the other HIVs, 2, 3, and 4, have their own unique events. So we're just talking about HIV-1 here, which we have the most evidence for. So what species did it come from? There is a related virus in monkeys called Simian immunodeficiency virus. Simian means monkey, SIV. And it causes a similar disease, destruction of the immune system. And SIV affects primates all over Africa, monkeys and apes, including chimpanzees, our closest genetic relatives. So a scientist traveled around Africa collecting fecal samples from wild primates because, luckily, if a primate is infected with SIV, virus is shed in the fecal material, so you don't need to catch wild primates in order to test them for the virus. You only need to find them up in the tree and then pick up the poop off the ground. So she traveled all over Africa collecting fecal samples from many species of monkeys and from chimpanzees in many different regions and gorillas. And it was found that, in fact, one population of chimpanzees in Cameroon, their SIV was by far the closest sequence match to HIV-1. And Cameroon, so here's Cameroon up here. This is a close-up of West Africa. And here's Kinshasa in the Democratic Republic of Congo. And Kinshasa is a very large city, and there's a lot of commerce in and out of Kinshasa. So is there any plausible way that the virus could have gotten from Kinshasa to Haiti? And in fact, there is. In the late 1950s, there were UN peacekeeping troops stationed in Kinshasa who were from Haiti, and they were there for several years, and then they went home. And that's probably how the virus got from Kinshasa to Haiti. And it's not hard at all to imagine how the virus got from Kinshasa to many other major cities in the region, because there's a lot of truck commerce and business travel back and forth between these different countries. So Kinshasa was the jumping off point for HIV-1 to the rest of the world. So how did that first person get it? The current hypothesis is what we call the cut hunter hypothesis. Because this has to be blood-to-blood transmission, somebody who was butchering a chimpanzee for meat, which happens all over the place in Africa. Chimpanzees and many other species of primates are butchered for meat. It's very easy to cut yourself when you're butchering an animal. So somebody was butchering a chimpanzee that was infected with SIV. They cut themselves. Some of the chimpanzee's blood got into their blood, and it was just close enough to the human receptor that this SIV was able to bind and enter human cells. And chimpanzees are very close to us. We're 98% similar from a DNA point of view, so that's not hard to imagine that something could jump that species barrier. There are many human viruses that can infect chimpanzees as well. So the cut hunter hypothesis is the most likely explanation. So sometime around 1908, somebody butchering a chimpanzee in Cameroon contracted the first human case of HIV-1. And then it slowly percolated through the human population, unnoticed, because the people who died from it would die of tuberculosis or pneumonia or something, and it wouldn't be recognized that they had a catastrophic collapse of their immune system because of the other obvious illness that they had. So there was another hypothesis that was disproven, and this was actually a hypothesis that the scientists had, was that possibly SIV was accidentally introduced to humans through vaccines, because we have to use live cells to grow viruses to make vaccines, especially the polio vaccine, which is a live virus vaccine. Green monkey kidneys were used to grow some of the early versions of the polio vaccine. So they were concerned. They thought, okay, is this from the green monkey? Could this virus have accidentally been introduced from monkeys into humans through the polio vaccine? And luckily, all the samples of the early versions of the vaccine, including, well, every vaccine, they're supposed to save frozen samples for future use, just for situations like this. So they tested all the early samples of the first polio vaccines that were grown on green monkey kidneys, and there was no HIV or SIV in those samples. So that is not where it came from. It's highly unlikely that it's was intentionally released into the human population, because before the discovery of HIV, it was not thought that retroviruses could cause disease in humans. So if somebody was developing some kind of a germ warfare agent, they would not have picked a retrovirus, because nobody thought that they would cause disease in humans. In fact, that was one of the surprising things with HIV, was that in fact we had a retrovirus, the very first one that was found to cause disease in humans, or any mammal for that matter. So if somebody, so that seems like it's very unlikely that it was some kind of a germ warfare thing. It appears to be completely wild, accidental viral introduction to our species.